All right, we're now talking about section 2, finishing up microscopic anatomy of skeletal muscle on muscle tissue for chapter 10 for anatomy physiology. And this is actually one of the final slides, but I still want to make that video last any longer. So now we're talking about how the mitochondria and other structures are associated with energy production. Muscle fibers have an abundant mitochondria, powerhouse of the cell for aerobic ATP production, affiliate part of the cellular transport chain. And basically, myoglobin within the cells allows for storage of oxygen that is used for aerobic ATP production, and glycogen is stored for when fuel is needed quickly for short periods. We'll talk more about that. Creatine phosphate can quickly give up its phosphate group to help replenish ATP supply. ATP lasting only basically 1 to 5 seconds. Creatine phosphate can give you energy for a couple seconds to about, some say 2 to 10 seconds, but really you could argue up to possibly up to 30 seconds. Glycogen kicks in somewhere around that 10 to 30 second period to start and becomes the predominant energy system until about 2 to 3 minutes, and ATP production is the predominant energy system. All of them still play a role to some extent, but the predominant energy system now for work periods that last 2 to 3 minutes or longer, more for endurance, right? And a lot of muscles need to have the ability to go for more than 2 or 3 minutes. That being said, innervation of skeletal muscle fibers. The motor unit is a motor neuron and all the muscle fibers it controls. So a motor neuron and all the fibers it controls is a motor unit. So we probably can say this without going too small, without going making it bigger here. Our spinal cord, basically sending and receiving feedback. You basically have our motor neuron here and the neuromuscular junctions. So basically the attachment where the neuron meets the muscle, basically the sarcolemma, that part of the muscle fibers themselves, to be able to fire the muscle fibers. And when it fires, they contract, they shorten. So whenever that signal comes, they shorten. That's their job, contract, right? The motor unit, the axons of the motor neurons come from the spinal cord or the brain. They innervate numerous muscle fibers. The number of fibers a neuron innervates varies. Small motor units have less than 5 muscle fibers. It allows for precise control of force output, whereas large motor units have thousands of muscle fibers, allow for production of large amounts of force, but not necessarily precise control. The fibers of a motor unit are dispersed throughout the muscle, not just in one clustered compartment. The synaptic knob on the neuromuscular junction is the expanded tip of the motor neuron axon. It houses the synaptic vesicles. These are small sacs that are filled with the neurotransmitter acetylcholine, ACH, and it has calcium, Ca2 plus pumps in the plasma membrane. It establishes the calcium gradient here with more outside the neuron. It has a voltage-gated calcium Ca2 plus channels in the membrane, and it flows into the cell down the concentration gradient if the channels are open. The motor end plate is a specialized region of sarcolemma with numerous folds. It has many acetylcholine receptors. The plasma membrane protein channels here open by binding of acetylcholine and allows the sodium entry and the potassium exit. Synaptic cleft is a narrow fluid-filled space that separates the synaptic knob from the motor end plate, and acetylcholinesterase resides here. It's obviously an enzyme, the ACE part, right? And it's an enzyme that breaks down acetylcholine molecules. So we have a neuromuscular junction here where the motor neuron innervates the muscle. It's usually the mid-region of the muscle fiber, has the following parts: synaptic knob, right here, the synaptic cleft, and the motor end plate. And our motor end plate here. Take a closer look here. So our neuromuscular junction occurring here, basically the axons, and down here the motor end plate are also known as a neuromuscular junction. The synaptic knob here. Nerve signal obviously being transmitted down through that to the end of that motor end plate to get the muscle fiber to fire. Synaptic cleft, or vesicle, excuse me, here. Synaptic cleft here, all through here. And basically the endomysium is the outer layer around the entire muscle itself, and a sarcolemma layer there, right? And then we have our myofilaments inside here, inside each myofibril. And then a close-up of our neuromuscular junction. The interstitial fluid outside the muscle cell, the cytosol, the cell itself. It has the sodium, it's going to be traveling out that sodium channel here, and the voltage-gated sodium channel, calcium channels here. I'm sorry, calcium. I said sodium. I said calcium. And also we have the voltage-gated calcium channels here. Okay, and the synaptic vesicle with acetylcholine. Our synaptic cleft here, and acetylcholine here. So obviously a negatively charged sodium here, the sarcolemma barrier, and here we have our acetylcholine receptor with the channel here, and we have our junction fold here, and this being the entire motor end plate. Or neuromuscular junction. The muscle fibers exhibit resting membrane potential, or RMP. The fluid inside the cell is negative compared to the fluid outside the cell. The resting membrane potential for muscle cells is about negative millivolts or megavolts. The RMP is established by leak channels and sodium potassium pumps. The voltage gated channels are closed. So here we have our neuromuscular junction. The skeletal muscle fiber here attached there to that neuromuscular junction for that neuron. Acetylcholine receptor going through here. Our voltage gated calcium channel. Then we have voltage gated potassium channels, and sodium here, calcium. I can't remember if I just said calcium or sodium. Calcium, we have basically our potassium and our sodium here, and there's our sarcolemma here with our myofilaments out of each myofibril. And the sarcoplasmic reticulum here, the T-tubules, the transverse tubules inside. So what did you learn? You should be able to identify the location and function of these connective tissue structures associated with muscle, the endomysium, the paramysium, the epimysium, the deep fascia, and superficial fascia. Draw and label a diagram of a sarcomere, and place the following gross anatomic and microscopic anatomic structures in order from largest to smallest: the fascicle, the myofibril, the myofilament, the muscle fiber, and sarcomere, and describe their anatomic relationship. Also, what's a motor unit? Why does it vary in size? Diagram and label the anatomic structures of a neuromuscular junction, and describe the distribution of the sodium channel and the potassium channel at the sarcolemma. And like so many times before, when you're having difficulty really understanding it, try to duplicate drawings and basically duplicate the diagrams yourself and make your own drawing, and then try to do it from memory without looking. Will play a great role in helping you understand this. For section 10.3, explain the events that lead to the release of the neurotransmitter acetylcholine from motor neuron. Describe the steps in excitation, contraction, coupling. Summarize the changes that occur within a sarcomere during contraction. Discuss what happens to each of the following to allow for skeletal muscle relaxation: acetylcholine, action potential, calcium concentration, sarcoplasm, and troponin-tropomyosin complex. Explain the relationship of skeletal muscle elasticity and muscle relaxation. So here we have our neuromuscular excitation of a skeletal muscle fiber. We have for that basically that neuromotor end plate, the neuromuscular junction here, right, where they connect here. And we have the synaptic vesicles that contain our acetylcholine. We have this action potential propagation that occurs here, basically at this neuromuscular junction, whereby we have the sarcolemma, the T-tubules, and the sarcoplasmic reticulum, and that excitation, contraction, coupling. The acetylcholine binding triggers the propagation of an action potential along the sarcolemma. Okay, and T-tubules to the sarcoplasmic reticulum, which is stimulated to release calcium. And that starts with, I should have mentioned, the release of acetylcholine from the synaptic vesicles and subsequent binding of ACH to ACH receptors right in here. And so we have that now, this stimulation, this signal that being sent down these T-tubules here to the sarcoplasmic reticulum, right? And then here you have the sarcomere with the crossbridge cycling. The calcium binds to troponin, triggering the sliding of thin filaments past the thick filaments, the thin actin filaments past the thick myosin filaments of sarcomeres. As sarcomeres shorten, the muscle contracts. Technically, the myosin heads are grabbing a hold of these actin strands and pulling, and it gives the appearance of muscle shortening with the muscles contracting and the fibers are being pulled toward one another, basically overlapping, giving the muscle greater size as it shortens, generally speaking. The calcium entry at the synaptic knob, the nerve signal travels down that axon, opens the voltage-gated calcium channels, calcium diffuses into the synaptic knob, and the calcium binds to proteins on the surface of the synaptic vesicles. Then you have the release of the acetylcholine from the synaptic knob. These vesicles merge with the cell membrane of the synaptic knob via exocytosis, and thousands of these acetylcholine molecules are released from about 300 vesicles. You want to know those exact numbers, but it's important to note that, generally speaking, there's a lot of them. The binding of the acetylcholine at the motor end plate is where the acetylcholine diffuses across the cleft, binds to the receptors, and excites the fiber. So we have this neuromuscular junction, the excitation of a skeletal muscle fiber, again down that motor neuron, that nerve signal, the synaptic knob here, and we have these voltage calcium channels here, our synaptic vesicles here, the acetylcholine released, and basically across that synaptic, travels across this synaptic cleft through the interstitial fluid there, and basically that motor end plate with the attachment point with a neuron meeting our muscle fiber here, or our muscle. There's an acetylcholine receptor here for that to happen in the first place. That nerve signal is propagated down that axon, and then it binds the synaptic vesicle membrane, and the calcium binding triggers the synaptic vesicles to merge the synaptic knob and the plasma membrane, and the ACH is exocytosed into the synaptic cleft, and that acetylcholine then diffuses across the fluid-filled synaptic cleft at the motor end plate to bind with these acetylcholine receptors. From a clinical view, Myasthenia gravis is an autoimmune disease. It's primarily in women. Antibodies that bind to the acetylcholine receptors and neuromuscular junctions. These receptors are removed from the muscle fiber by endocytosis, and they're resulting in decreased muscle stimulation with rapid fatigue, muscle weakness. Eye and facial muscles are often involved first, and it can be followed by swallowing problems and limb weakness. And like so many illnesses, ailments, diseases, disorders, syndromes, there's always fatigue is a common one. There's always fatigue, weakness, and potentially some sort of pain. Certainly there's dysfunction, but the intricacy of all the numbers of functions are what make it so challenging at times to figure out where the cause or where the problem really lies, especially when it starts as something as simple as fatigue and weakness. But that being said, these are the indicative factors of myasthenia gravis. For the excitation contraction coupling, stimulation of the fibers coupled with the sliding of these filaments, thick and thin filaments, actin and myosin. The coupling includes the end plate potential, EPP, the muscle action potential, and the release of calcium from the sarcoplasmic reticulum. This one was a little bit smaller than I intended with this slide here. It's still too small. It's even with increasing the size. I don't know if I can get it big enough now. I'm not going to be able to. We're going to skip past this. It's just way too small to try to make this clear for you. We need a better slide here. I should have got a better slide for you. This will give us a little bit more of it here. That said, the action potential propagation here, traveling down that T-tubule, again, our voltage-gated potassium channels and voltage-gated sodium channels, and we have the release of this calcium following. Well, it was the first step we couldn't see very well, but we'll come back to that. The action potential then propagated along these T-tubules and to stimulate this voltage-sensitive calcium channels, which then trigger the opening of the calcium release, which is located in the terminal cisternae of the sarcoplasmic reticulum here. Okay, and it diffuses out of the cisternae and into the reticulum within the cytosol. The implant potential is when the ACH receptors are chemically gated channels that are open when the acetylcholine binds to them. Sodium diffuses into the cell through the channels, while a little potassium diffuses out. And the cell membrane cell membranes briefly becomes less negative at the end plate region. And that end plate potential is local, but it does lead to the opening of voltage-gated ion channels in the adjacent region of the sarcolemma. And that leads to the initiation, propagation, action potential along the sarcolemma and the T-tubules. This action potential, or AP, is a rapid rise, also known as depolarization. It's going to come back a lot. And fall, the repolarization in the charge of the membrane. And EPP reaches its threshold by causing nearby voltage-gated sodium channels to open. And the sodium diffuses into the cell through these voltage-gated channels. And the cell depolarizes, meaning it becomes less negative, and eventually becomes plus 30. This results in the opening of adjacent voltage-gated sodium channels and more sodium entry. And then chain reaction occurs as depolarization is propagated down the membrane and the T-tubules. And then initiation and propagation action potential continues with the sodium channels opening. They close and voltage-gated potassium channels open. The potassium diffuses out of the cell repolarizes, returns to -90, and the repolarization is then propagated down the membrane and the T-tubules. And while the cell is depolarizing and repolarizing, it's in a refractory period, unable to respond to the stimulation. So we have basically that threshold here, where with a 90 here, where we actually don't have any activity here. That threshold points -65. You see depolarization occurring, and the repolarization is going to return and bring us back down after it gets as high as +30, back down toward -90. And it all happens in basically a couple of milliseconds, extreme short period of time for this to happen. That said, that sarcolemma, the unstimulated skeletal muscle fiber, has a resting membrane potential. The RMP is negative 90. The threshold or end plate potential is reached when those acetylcholine receptors are those chemically gated ion channels open, and there's then sufficient sodium enters the motor end plate to change the resting membrane potential from negative 90 to negative 65. That's the threshold value we're looking for. Then depolarization occurs at negative 65 as these voltage-gated sodium channels on the sarcolemma open up, and the sodium enters rapidly. That reverses the polarity, getting it back to negative 65 down to plus 30, or up to plus 30, I should say, to a positive plus 30. Then repolarization occurs due to the closure of these voltage-gated sodium channels and the opening of the voltage-gated potassium channels on the sarcolemma. And then the sodium moves out of the, or potassium, excuse me, moves out of the cell, and the polarity is reversed from positive to negative, positive 30 to negative 90, returning back to our resting membrane potential. And then the action potential travels down these T-tubules, causes the voltage-sensitive calcium channels in the T-tubule, the membrane, to trigger the opening of the calcium release, and that release results in this sarcoplasmic reticulum terminal cisternae is where it reaches, and then the release of the calcium from the sarcoplasmic reticulum causes that calcium to interact with the myofilaments, triggering contraction. And I think we're going to go ahead and stop at this point right here. We'll talk more about Sarcomere and Crossbridge cycling here in a moment.